Hello, my name is Ryoya. I'm the S2 here at ICT. So, I would like to introduce my research that I did this summer 2023. So, first of all, do you guys know what Maitake-Protease is? So, I think most of you know what Maitake is. So, I wrote here Maitake-Mushroom. So, Maitake-Mushroom, as most of you know, Maitake-Mushroom is kind of an edible mushroom sold in supermarkets. It's very cheap. It cultivates in Japan and China. It's not really rare. It's a really common mushroom in Asia. And so the reason why I chose to do research about Maitake is that Maitake is known as one of the mushrooms that contains a lot of protease. Protease is an enzyme that breaks down proteins into amino acids. So I've heard from my sister that if you add maitake, a raw maitake, to a dish, for example, called chawanmushi, they use this egg to make like an egg pudding. By using the raw maitake, by cooking it, it's gonna not be fixed like a jelly. It's gonna break down and become like a juice. And so I got interested in maitake mushroom, and so I started researching about it. And the reason I would like to research about maitake, I was reading a lot of researches about maitake mushroom, and I found that no one has ever done research about maitake and other mushrooms like normal brown mushrooms and white mushrooms. I found researchers finding differences between Japanese maitake and other white maitakes, or shiitake or bunashimeji and other mushrooms that cultivates in Japan, but I never saw comparing mushrooms, so Western mushrooms and Japanese maitake. So I did an experiment to find differences between those two. The purpose of this experiment is to investigate proteolysis ability in different conditions. So I made some few different conditions to compare. So those are raw mushrooms, and dividing them into two parts, the cap and the stem. And the other one is the whole thing, mixing all them together. And the other one is defrosting them. So the reason why I defrosted them is that by freezing them and defrosting them, the protein, the protease is also a protein. So by freezing, you can break down the enzyme itself, and so that I can dysfunction the enzyme. And the hypothesis that I made is to proteolysis cap to stem ratio. So the reason why I divide them into cap and stem, that I wanted to know if there's differences between the cap and stem, the amount of proteolysis ability. So that's differences between just one whole thing, raw mushroom, and freezing and defrosted mushroom. And so the method that I did is to, number one, I made an agar media, but actually at first I made a milk media, but it didn't work well. The reason why milk media using a gelatin, it didn't really work well. The reason why is that by using gelatin needs a condition that it's cool in order to be fixed. But for this maitake mushroom protease, by reading the researchers, I found that it works really best in like about 60 degrees to 70 degrees Celsius. So if I added heat, the gelatin broken down and it became a liquid, and I couldn't really find whether the amount of gelatin that had broken down is the amount of gelatin that had broken down by the enzyme or by the heat. So I had to add some kind of changes to the media, and I chose agar because it's really has a high resistance to heat. And but there was a problem. So I used gelatin first, so that's why I can have protein to break down. But because I used agar doesn't have any protein inside, so I had to add kind of a source of protein. And so I chose milk and soy milk as a source of protein, using one of the milk is the animal-based and soy milk is plant-based. And number 2, adding added mushrooms to each cup. I added about 2.5 mushrooms that I cut down into pieces so that I can increase the surface area so that I can have more ability to break down proteins. Number 3, keep the temperature around 6 degrees. As I said, Maitake-Mushroom Protease works very good, the best efficiency in around 60 degrees Celsius. So I chose that temperature from the other researchers. And number 4, measure the pH level and protease solubility. So the reason why I chose pH level is that by having low pH, it'll help break down proteins better. So I was looking for if there's any changes between the normal brown mushroom and brown maitake in pH, and it's really difficult to see, but when you use raw mushrooms, raw maitake, and adding heat with protein, the protein breaks down and becomes like a liquid. So I was able to collect the amount of liquefied proteins, and so that I was able to measure the protease stability. So the result, I said I used milk and soy milk, so I have 2 graphs here, and each of them are cap, stem, whole, defrosted, and a control. So here is really a rough illustration picture here. I used 9 of them because I used a brown mushroom and maitake for each of them. So I had 4 conditions and 1 control, so 9 of them. And so the most important thing is that most important proteolysis ability activity in maitake was localized in the cap, while for brown mushroom it was localized in the stem. So as you can see here, maitake had our highest proteolysis ability here in the cap, the blue graph. And for brown mushroom, the gray graph, you can see that the stem has more proteolysis ability than the cap. And as a whole, maitake had less than the brown mushroom. And for defrosted, there was more proteolysis ability in maitake than brown mushroom, but also the amount of proteolysis ability had decreased from the raw whole mushroom. And there was one problem that first I did the milk media experiment, and so I used heating machine to heat really evenly, but using hot water. But by using hot water, the water that comes up, because I used a lid to keep the temperature, the steam condensed and became liquid water and dropped down to these cups. And so there was a lot of liquid inside the control, which had added nothing, and I got 9.2 grams. And so I counted all of them as equal condition, and I subtracted 2.5 grams for the accounting for the amount of mushrooms that I added initially, and the amount of liquid that I got for control. And those are the orange and yellow graphs. And those are really the same because I'm decreasing the same amount. And for soy milk, you can see that it's almost the same, but maitake had a higher in cap and brown mushroom had higher in stem. But also, if you look at the whole raw mushroom, maitake has much higher amount than brown mushroom. And because I made a mistake in milk experiment, I made an aluminum foil lid in between the lid and the actual metal lid and the cups. And so that I was able to prevent a lot of those condensed steam to drop down into the cup. And so I only got 0.3 grams in the control. And I did the same thing, decreasing the amount from the data, the amount of liquid from the control, and 2.5 grams accounting for the initial amount of mushrooms. And those are orange and yellow, and those are almost the same, but you can see that the whole mushroom, brown mushroom, it was almost 0 for whole, and defrosted was almost 0. So it means it was more difficult for the maitake, both maitake and brown mushroom, to break down soy milk than milk. And also for the data, maitake, for example, maitake cap broken down about 6.3 grams, and for soy milk, they only broken down 2.3 grams. That's only about like 1/3. And so, as you can see here, there's less bias here in soy milk. So I think I can assume the soy milk experiment has more accurate data, and from that data you can see that maitake has more proteolysis ability than brown mushroom. And so by defrosting them, all the data has decreased. So I was not able to dysfunction all of the functions for the protease, but it has decreased the amount of proteolysis ability. And so, as I said, I did measure the pH change, and for raw maitake, it was about 4, and brown mushroom was about 5 to 6. And so, those all had our lower pH in 7, and so that's a bit acidic, and I think that helped maitake break down more than the brown mushroom. But because I made a mistake here in milk, I can't really actually say it's the pH that helped break down more protein. And for temperature, I kept, like you can see here in the graph, I was keeping the temperature around 60 degrees Celsius, and I did for 900 minutes of 15 hours. And the reason why is that from past researchers found that the maitake protease works about for 8 hours. And so I took about double the time because I wanted to see if the brown mushroom have different types of protease. And from here you can say that these all had, even with the same amount, they had different proteolysis ability. And so you can say that maitake and brown mushroom have different types of protease, and maitake has more proteolysis ability. Thank you very much.